mice already. They have a big problem with inmates down in the Bakersville, Fresno area that have HIV/AIDS. They get continuity exhibits, and it's devastating. Yeah. It's really hard, really bad. And we need to know about it here in California, you guys, because you may have been exposed already. And the old lesions might look like cancer. So if you have a radiograph later, you guys say, oh, could we see a suspicious shadow in your lungs? It could be cancer. Tell them you lived in California, and they need to make sure it's not an old valley fever lesion. Otherwise, they end up taking part of your lung out. That's happened. So remember that, you guys. You've been exposed to valley fever. Okay, guys. Those guys are awesome. Okay, so you guys, the last one then of our lipid are the waxes. And again, you're like, what the heck? Why are we talking about waxes? So you guys, there's a big group of pathogens which belong to the Mycobacterium genus, and there's two species that cause really serious disease, Mycobacterium tuberculosis. And what do you think that causes? Tuberculosis, right? Big problem here in California. And then its cousin is Mycobacterium leprae. And which disease do you think Mycobacterium leprae causes? Right. And you guys, it's really important that if you're working with a patient that's infected with Mycobacterium leprae, you don't tell them they have leprosy. You tell them they have Hansen's disease, a bacterial infection. And the reason for this is, you guys, is that if my doctor came in and said, you have leprosy, emotionally I think, oh my gosh, I'm no longer human, I'm a leopard, right? So there's all this historical, emotional baggage that goes along with being told you have leprosy. So again, when talking with the patient, family, and friends, you say, you have a bacterial disease called Hansen's disease, and you try to avoid the term leprosy, right? Because you don't want them to suffer more. And you need to tell them it can't be treated, right? It is bacterial. But you guys, we have a big problem with both Mycobacterium tuberculosis and Mycobacterium leprae, and that is they have a very hydrophobic, lipid-rich, waxy cell wall. So they have these incredibly hydrophobic waxes in their cell wall. And our example is mycolic acid. And mycolic acid, you guys, it has these hydrocarbon tails that could be 20, 30, 40 carbons long. So it's just so water resistant. And that's a problem, you guys, because our antibiotics are usually water soluble, right? They're hydrophilic. Our staining reagents, like we're going to do gram stains next week, they're water soluble. Nutrients are usually hydrophilic, right? So all of these water-soluble hydrophilic substances have a hard time diffusing across the cell wall of the mycobacteria. Because of the waxy cell wall, they're really slow growing, so it slows down diagnosis. You can't grow them very quickly. It slows down antibiotic sensitivity testing, right? If I'm coughing out mycobacterium, you guys, this waxy cell wall prevents them from drying out, so they're going to remain resistant. They're going to remain infectious in the environment for long periods of time. They're resistant to a lot of antiseptics, a lot of disinfectants, and furthermore, you guys, that waxy cell walls prevents them from being killed by our host phagocytes. It's just really hard for our phagocytes to kill them. So these guys are tough, right? They've got the most resistant cell wall of all. And because, you guys, it's hard to gram stain them, they had to develop a special staining procedure that we'll talk about in lab next week. We aren't going to do it because it's kind of a toxic protocol. But the special staining procedure, you guys, is called the acid-fast stain. Acid-fast meaning that once you stain the mycobacterium and you rinse them in acid, hydrochloric acid, the cell wall is so tough it won't break down and the cells retain the stains. All other cells, the acid breaks down the cell wall or cell membrane, and everybody else becomes colorless. And so these bacteria, guys, are called acid-fast bacteria. So right off the bat, if we hear acid-fast bacteria, we're thinking of a waxy cell wall. And sometimes, you guys, they're abbreviated as AFBs for acid-fast bacteria. So right off the bat, I think of mycobacterium. Yeah. So Another problem with this, you guys, is because so many antibiotics can't diffuse across the cell wall, you have to be really careful choosing the antibiotic you use to treat your patient, right? And another complication is sometimes the patient has to be on the antibiotic for months, sometimes years, and sometimes they get tired of taking antibiotics. One of the frontline anti-TB drugs you guys, so I'll put this for treatment, it's called isoniazid, or INH. This is a frontline anti-TB drug. And this is really cool, you guys, because what it does is it inhibits, it stops mycolic acid synthesis. Right? So you're going to decrease production of those hydrophobic waxes in the cell wall. And then you always have combination therapy. You never give just like one drug, you always have 2 or 3 or maybe 4 drugs on board. And so to me, it's like, oh, that's so cool because the isoniazid is going to decrease the wax production, and therefore the second antibiotic that you're giving the patient has a better chance of diffusing across the cell wall to actually help kill the mycobacterium. Okay. So again, you guys, next week in lab, we'll be mentioning the acid fasting, and we'll talk about the reagents and how mycobacterium would appear, and how non-mycobacterial cells would appear. Okay. All right, folks. So I think that's it. I think we're finished there. There's a few additional slides you guys, and yes, me just being kind of nerdy, so you can ignore those.